Hi, in this short tutorial, I'll be talking about vitamins and minerals as coenzymes and cofactors. In order for us to do so and have an understanding, we need to refresh our minds with what exactly enzymes are. So what exactly are enzymes? They are biological catalysts that fasten the rate of biochemical reactions within the biological systems. You can say that enzymes are catalysts within the biological system. Now, enzymes speed up biochemical reactions, but they are not used up or changed in the process. And so an enzyme can function over and over again. It's important to also remember that most enzymes are proteins and they consist of one or more polypeptides. And you will also remember that proteins are made of amino acids in various sequences. However, there are a few enzymes that are non-proteins. An example of these are ribozymes. We have Hovnik and hammerhead ribozymes, which are found in humans. Besides this, most of the other enzymes are essentially proteins. Now, in order to understand cofactors and coenzymes, we will mention the concept of enzyme helpers. Now, both cofactors as well as coenzymes are essentially molecules that facilitate enzymatic processes. In other words, their activity is limited in the absence of cofactors and coenzymes, and that's the reason why they have their names. They are co-players or co-partners in the enzymatic processes. It's important to understand and remember also that there are enzymes that function without this assistance of these enzyme helpers. An example of these are pepsin and ribonuclease. They do not need cofactors or coenzymes. Now back to our main subject of vitamins. Vitamins and minerals are substances that are required in small quantities within the biological system, and so they are important either as building blocks or they are part of metabolic enzymatic processes. Let's talk about cofactors. Cofactors are essentially inorganic ions such as magnesium, iron, zinc, copper, and these are necessary for the function of enzymes. Now remember that enzymes are essentially protein in nature. However, these cofactors are non-protein molecules, so the non-protein molecules assist the protein molecule in their enzymatic processes within the biological system. Now, cofactors help in electron transfer during biochemical reactions. Now, this electron transfer is important for the function and activity of the enzymes. Again, don't forget that most enzymes are proteins, which are essentially amino acids. And so you have a non-protein component facilitating the action of a protein enzyme. Cofactors are not as complex as the protein enzymes, but because they are not protein, they are less sensitive to heat. In other words, they are not as thermolabile as the protein enzyme. And so they tend to increase both the structural as well as the thermal stability of enzymes, which leads to facilitation of their action. Cofactors mostly bind to allosteric sites on the enzyme and alter the enzyme structure to facilitate binding of the substrate and the enzyme. Now, the allosteric site is a site that is outside the active site of the enzyme. So they may donate chemical groups that help stabilize the structure as well as the activity of the enzyme because of the fact that they are able to bring stability to the structure as well as the active site. Now, cofactors may also bind loosely, in other words, not like a kind of strong covalent bond like most people would tend to think. So they may bind loosely and be able to dissociate easily. Now, an example of a cofactor is the case where magnesium ions are necessary for the DNA polymerase, which is involved in enzymatic processes that lead to production of DNA. So, in the absence of magnesium, the DNA polymerase activity is impaired. In fact, magnesium is an important cofactor for the addition of nucleotides to the growing DNA strand. Now, to prove this, it has been shown that when running a polymerase chain reaction, the absence of magnesium ion or the insufficiency will lead to no product. Zinc is a cofactor for superoxide dismutase. This is an enzyme which also has an antioxidant effect. So, without the presence of zinc, the effect of SOD will be, to an extent, diminished. SOD is an example of a metalloenzyme. In other words, it requires zinc to function. That's why it's called metalloenzyme. Copper is an important cofactor for cytochrome oxidase, and zinc is an important cofactor for alcohol dehydrogenase. Then magnesium also for ATPase enzymes. Now let's quickly look at coenzymes. Now most coenzymes are either vitamins or they are derived from vitamins. In other words, coenzymes are actually organic in nature, meaning that they have a carbon component. Now, since vitamins are organic, coenzymes are usually organic. That's just basically what it means. Because they are organic, it means coenzymes. So you can say that while cofactors are inorganic in nature, coenzymes are usually organic in nature. So coenzymes assist enzymes by ensuring the substrate is well attached to the active site. So a moment ago I talked about the cofactors. Most often than not, cofactors tend to bind to allosteric sites, while coenzymes usually will bind within the active site. So coenzymes also help in stabilizing some substrates and also transfer important functional groups needed for the reactions. Coenzymes can be part of the enzymatic reaction because they provide a functional group that is part of the reaction. So they have a component that they donate or a component that is important for that metabolic activity to take place. So example of some of the functional groups that a coenzyme may donate or make available as a component of its own structure are methyl groups and acyl groups. So examples of coenzymes are thiamine, which is from vitamin B1. It's a coenzyme that helps in body metabolism of carbohydrates to yield energy in the form of adenosine triphosphate. We're all familiar with ATP. So this coenzyme is important in the formation of ATP. Vitamin K is also a coenzyme that is important for vitamin K dependent carboxylase. Now, this enzyme is necessary for the synthesis of proteins involved in hemostasis. So, for blood clotting to take place, you need this vitamin, which is a coenzyme. Niacin is a source of nicotinamide adenine dinucleotide, and this is a coenzyme for alcohol dehydrogenase. Now, as a caricature, you have the yellow being the cofactor binding to the allosteric site, the substrate binds to the active site, the reaction takes place, the product is formed, and you have the release of the cofactor. For coenzymes, the coenzyme binds in the active site and also the substrate. The reaction is facilitated, the product is made, and the cofactor can be recycled. By way of recap, we can say cofactors and coenzymes help facilitate enzymatic reactions within the body. Both are non-protein molecules. Cofactors are largely inorganic and are found in minerals, while coenzymes are organic in nature and from vitamins or derived from vitamins. Coenzymes mostly bind at the enzyme active site, while cofactors may bind to allosteric sites.